Welcome to This Explainer. Today, we're tackling a massive, fundamental scientific problem. Think about it for a second. Human bodies are constantly reacting to messy, noisy, completely unpredictable environments. I mean, we drink coffee, we get stressed out at work, we lose sleep, we eat weird foods. So when researchers are testing a medicine, how on earth do they definitively prove that the medicine actually caused a specific symptom rather than it just being a result of all that everyday physiological noise? Well, we're going to unpack the rigorous methodological workflow of homeopathic pathogenetic trials to see exactly how they solve this massive puzzle.
All right, let's take a quick look at today's roadmap. We're going to start with the quest for pure data. Then we'll move into controlling the background noise, look at the art of neutral observation, explore inclusive and safe trial design, and finally, wrap up by comparing preparation types. Let's jump right in.
Section 1. The quest for pure data. Isolating true medicinal effects from everyday physiology. So, looking at aphorism 124, it highlights this brilliantly practical philosophy. And what's absolutely vital to grasp here is that the goal of a proving trial is not ascetic deprivation. Forcing a participant into some radical, highly restrictive diet, like suddenly fasting or completely dropping all carbs, that actually creates massive physiological stress and withdrawal symptoms. And honestly, that metabolic noise would just completely ruin the proving record. Instead, the real aim is proportionate control. It's about keeping ordinary routines stable and just eliminating those avoidable, unusual influences that can mask the true pathogenic effects we're looking for.
Moving on to Section 2, controlling the background noise, establishing a stable baseline and regimen. Now, this 7 to 14-day baseline run-in phase is absolutely fascinating. Before a participant, or as they're known, a prover, ever takes a single drop of medicine, they have to meticulously track their natural state. They're logging their usual headaches, sleep duration, bowel habits, stress levels, the whole shebang. And this gives researchers a highly detailed, incredibly reliable map of what is physiologically normal for that specific person. Like if a prover regularly gets a tension headache on Thursdays because of academic stress, this baseline catches it. That ensures investigators don't falsely attribute that pre-existing headache to the new medicine later on down the line.
So we absolutely must proportionally control everyday disruptors. Just think about your own daily habits for a second. A sudden caffeine binge? That can cause tachycardia and insomnia. Sleep deprivation from a crazy work week? Hello, frontal headaches and cognitive fatigue. And radical diet changes usually mean bloating and massive energy crashes. If a prover experiences these kinds of exposures during a trial, those symptoms mimic the potential effects of a drug. And well, that just completely trashes our data integrity. Because of this, the protocol demands that provers maintain a super stable, habitual intake. No weird new dietary supplements, definitely no extreme fasting, and for sure, no sudden late-night espresso shots.
All right, Section 3, the art of neutral observation, protecting the prover's authentic account.
There is this incredibly sharp contrast in how supervisors conduct daily debriefs. If a supervisor asks a leading question, something like, did your headache get worse at 3 p.m.? Or did the dose cause burning in your stomach? They are artificially planting expectations. They are literally cueing confirmation bias and manufacturing artificial remedy keynotes out of thin air. To prevent this kind of suggestive bias, supervisors are strictly trained to use only neutral prompts. Things like, what changed today? Or when did it begin? This approach is absolutely crucial because it protects the purity of the prover's authentic subjective experience, letting them describe what's happening in their own unpolluted language.
But let's be real here. Participants are human. So what happens when a prover inevitably breaks the rules? Say they chug 4 cups of coffee during exam week and then report heart palpitations. Well, adherence is measured. It's not just assumed. Supervisors don't just guess or immediately throw the whole trial in the trash. Instead, they use a very precise, totally transparent action pathway. First up, they accurately log the exact timing and nature of the deviation. Second, they formally assess the impact, like does that coffee directly explain the palpitations? And finally, they firmly adjudicate the event. They might flag the data, pause dosing until the baseline stabilizes, or in really severe cases, withdraw the participant entirely just to save the integrity of the data.
Which brings us to Section 4, inclusive and safe trial design, expanding relevance while protecting participants.
Hahnemann explicitly stated that medicines must be proved on individuals of different sexes. And the massive takeaway here is that failing to test on different sexes and genders leaves these huge demographic blind spots in the Materia Medica. Think about it. If a trial only recruits young men just to simplify the study, researchers are going to completely miss sex-specific biological symptoms like alterations in menses or unique hormonal patterns. And an incomplete proving record, that ultimately leads to failed clinical prescriptions for diverse patient populations. But of course, testing a broader population brings profound ethical and design responsibilities. For instance,
If a female participant, let's use the source case study of Provero 8, takes a home pregnancy test on day 5 of the trial and it comes back positive, a strict reproductive safety protocol is instantly triggered. Fetal safety always supersedes data collection. This launches 3 mandatory steps: immediate dosing cessation, a controlled emergency unblinding by the medical safety lead to check for any toxicological risks, and swift regulatory notification. Furthermore, any symptoms she reports from that point, like morning nausea or suddenly being sensitive to smells, they have to be excluded from the trial data. Why? Because they're physiological hallmarks of early pregnancy, not the test medicine.
Now for our final section, Section 5, comparing preparation types, crude versus potentized arms. Aphorisms 128 and 129 outline a pretty stark difference between trial arms. Crude preparation trials, they use measurable chemical doses. They're tracking gross material toxicity and objective physical signs. And because of the high toxicological risk there, they require extremely strict safety limits. On the flip side, potentized preparation trials use ultra-diluted substances. They have a very, very low material toxicity risk, and instead, they track these incredibly fine mental, emotional, and highly subjective functional shifts.
But here's the catch, right? Because those potentized remedies are designed to be so incredibly subtle, how do we actually know the prover isn't just imagining things? Well, we rely on credible double-blind placebo controls as the ultimate lie detector. By keeping both the participant and the supervisor completely in the dark about who has the active medicine and who has the placebo, researchers can accurately measure that background noise and psychological expectancy. If the symptom frequency in the active arm is basically equal to the placebo arm, then the substance showed no discernible effect. It's a brilliant way to completely separate a true dynamic medicinal effect from the simple power of suggestion. So we've seen exactly how researchers have to rigidly govern diet, totally eliminate suggestive bias, enforce strict inclusion protocols, and utilize placebo controls, all just to identify a single valid medicinal symptom. To wrap up this explainer, I want to leave you with a thought to really challenge your own perceptions of health and data. Considering all the physiological disruptors in our modern world, from the stress of your morning commute to that extra shot of espresso you had at lunch, until you establish a completely stable baseline, can you ever really trust what your body is telling you?